Hello, everyone. I am Luiza Albuquerque from Fluminense Federal University in Rio de Janeiro, and I'm glad to welcome you to this ALAGO webinar. ALAGO teams is working very hard to stretch the bounds of our scientific community. However, it would not be possible without the support of our sponsors. So I would like to thank FAPERJ, SEMS, Nova Analytica, and Termo Scientifics for all your support, as well as I would like to thank the support of the young scientists that very efficient, the technical support committee. So some important notes before we start. Please don't forget to follow us on the YouTube channel and in our social networks. And last but never least, I would like to invite all of you to join our association, and the link is available in the description of this video. Finally, starting to present now our speaker today. As you know, organic geochemistry is a very powerful tool for many things, including paleoenvironmental reconstructions. And in the last years, more specific techniques as stable isotopes applied for specific compounds have been applied very successfully for a more accurate and complex inference. This is the general idea for what our invited speaker today will talk about today. Then I would like to introduce our speaker today is Dr. Susanne Alfken, that is a prominent young scientist that built her career on the University of Bremen in Germany, and now is a postdoc at MARUM. Is applying her knowledge on organic proxies to reconstruct the paleoclimate. Thank you so much, Susanne. Please welcome.
Thank you. Thank you so much for this very nice introduction. So should I start the presentation mode now?
Tommy?
Okay. Yes, you can start it, Professor.
Okay, I can start it. Thank you. So I hope you see my presentation. And I was told I should use the laser pointer. So again, thank you very much for this very nice introduction. And of course, a very big thank you to the organizing committee of this nice webinar, especially Vinicius, who asked me to present our approach of high-resolution paleoclimatology via mass spectrometry imaging of molecular proxies in sedimentary archives. And this project is funded or was funded by the European Research Council, which was an advanced grant, and we are just finalizing the final report. So the whole funding period is over at the moment, but we will continue with this method and with this approach at the MARUM at the University of Bremen in Germany. So when we think about climate change, we usually like to look at the recent temperature increase in the past century, in which we see that, especially after the 1970s, we have this massive temperature increase. But how do we know that this temperature increase is normal or is maybe due to anthropogenic reasons? And one good way to see what happens today is actually to compare it to what we saw and what we know from the past. And when we do this for the past 2,000 years, we actually see that this warming rate we observe in this century is much, much higher than anything else we can observe during the past 2,000 years. But where does actually this reconstructed data come from? And for this, we have quite luckily a variety of climate archives which we can use. For example, we can use tree rings from the terrestrial side, but we can also look into corals, which give us information about temperature change and climate and environmental factors, as well as ice cores from the glacial ice. And one archive that we are using in the MARUM, and probably a lot of people everywhere else too, are marine sediments. And within these marine sediments, I like to look into molecular stratigraphy and use molecular stratigraphy on these marine sediments to address climate change. And this was quite a new tool in 1986, but has so far become quite regularly used. And to give you a short overview, what it's based on, for those of you who are not so familiar with molecular stratigraphy, is that when we have here our ocean basin as a sketch, it's not to scale everything. You can see that we have some vegetation on the side, which might, for example, burn. We have our primary producers in the upper ocean. And then we have microbes, bacteria, archaea, actually all over the water column. And what these organisms or these processes all have in common is that they have specific biomarkers or produce specific biomarkers. For example, we have these long-chain alkanes, which are in the plant leaf waxes of these land plants. Or when we burn biomass, we create these monosaccharide anhydrates, it's levoglucosan. Or we find these long-chain alkenones in the coccolithophores in the water column, as well as dinosterol from dinoflagellates, or these isoprenoidal tetraethers in the taumakia in the water column. So when all of this material that is in the water column and on the land is transported via sedimentation to the ocean seafloor, we still find these biomarkers in the sediment over time. So when we come along with our research vessel and drill into this sediment and take out a sediment core, what we usually do in molecular stratigraphy is that we take out about centimeter-sized samples and we extract them with organic solvents to get out our total lipid extract, which we can then further analyze with, for example, HPLC-MS or GC-MS to get an idea on our compounds and to separate them in there. So we have our chromatogram with the relative intensity of our retention time, and then we can separate and see, for example, our alkenones eluting up here and the isoprenoid tetraethers eluting much later. And then we can use, for example, these alkenones to plot them and to look at them over the sediment core. So from the top part, from the young part, down to the bottom part, to the older part. And then we get, for example, a record of these alkenones, which are representative of sea surface temperature over time. And we see some variation in there. But what we need to acknowledge and think about with this record is that one data point actually consists of several millimeters to centimeters of sediment. So transferred into time, and depending on the sedimentation rate, this means that we have several decades up to centuries combined into one data point. So this means we actually lose a lot of information due to this high sample demand. So we thought about how to improve the resolution and how to overcome this loss of information due to this high sample demand. And we came up with the idea of using an approach that is called mass spectrometry imaging. And with mass spectrometry imaging, what we do is that when we have our sediment core, we take out with these double L channels intact parts from the sediment and divide it into 5 centimeter pieces, which are then thinly sliced with a cryomicrotome. And then we shoot with a laser onto the sediment at a defined raster with about 200 micrometer resolution from spot to spot. And this impact of the laser leads to a desorption and ionization of the compounds, which are transferred to the FTICMS, so a very high resolution and powerful mass spectrometer. And with this, we can then actually, when we have here our sediment slice, visualize the UK37, so the proxy distribution on the surface of the sediment. So when we compare these two approaches, we can see when the conventional approach that we have a resolution from about decades to centuries. With mass spectrometry imaging, we can now actually visualize, so image the biomarker distribution. And when we correct for these slightly tilted laminations in there, which are in this piece due to the sampling and due to the core structure, we can then average along every horizontal line and get a 200 micrometer down core resolution profile, which translates into about annual to monthly resolution. And when we compare these two records, we actually also see that we have a very strong correspondence between the extraction, so conventional approach, and this new laser-based analysis. And all of this is put into a whole sample preparation pipeline. So when we start with our sediment core and we sample it with a double L channel and take out these 5 centimeter pieces, which we freeze dry and embed it to make it stable again. And then we actually ended up with two versions. And if we want to combine the whole part with an elemental mapping, we need a bit thicker slices of about 100 micrometer to get good results from the elemental mapping and the biomarkers. But if we only go for the biomarker analysis, thinner slices are a bit more ideal for the signal intensity. And to give you an idea how this looks like, you can see here these embedded sediment pieces in the gelatine, so these little cakes, so to say. And over here you see in the picture, when we freeze these sediment pieces in the gelatine, we can then use this cryomicrotome and get out these very thin slices from the sediment. And what I just want to point out in this picture as well is that we still see the nice structure in the sediment. So this was a bioturbated test sample, and we still see these round-shaped structure over here. So we don't disturb and screw up the whole structure in the sediment by this whole sample preparation process. And all of this is already published in Organic Geochemistry. So if you want to get the full recipe of how to prepare samples for mass spectrometry imaging, you can read it. It's open access. And yes, just read it if you like. So now that we have this sample preparation process and the whole pipeline set up, we wanted to look in what kind of biomarkers we can actually measure. And this is just some examples of the biomarkers we were able to detect. There are some more, but what I want to point out is that we can look into sea surface temperature based proxies or based biomarkers that we can use to reconstruct sea surface temperature, such as these long chain alkenones or the isoprenoidal tetraethers. We can get an idea of the marine versus the terrestrial input by using the fatty acids, with the short chain fatty acids C16, C18, being more indicative of the algae signal, whereas the long chain fatty acids stand for the terrestrial input. The same can be done with the sterols, with the C27 and C28 sterols also having a stronger signal from the algae part, whereas the C29 sterols derive from the vascular land plants. And the sterols are also nicely, or can be nicely used to get an idea about the redox conditions in terms of oxygen availability, meaning that we have this stenol to stanol conversion at the oxic-anoxic interface, which is then indicative of the oxygen conditions. So meaning when we have more oxygen, we find more of this stenol, whereas when we have less oxygen, you can detect more of the 5-alpha cholestanol in this case. Furthermore, we can look and investigate the bacterial community composition by detecting the BHPs, or also look into the primary production signal with a variety of pigments that we were able to detect, and also the degradation products. And if you want to get also more detail on and more insights into the compounds we can detect and how we were detecting them, this is also published in Organic Geochemistry and also open access. So always easy to get a look into what we are doing at the moment. And one major issue or one major context I want to point out is that it's very extremely important that we have this very high mass resolution. This is why we use the FTI-CRMS, because we don't have a chromatographic separation of our compounds. We just rely on the mass spectra. And with the FTI-CRMS, we're actually able to separate, for example, here with the GDGTs, the isotopologues, meaning when we have our GDGT1 over here, we can separate it from the GDGT2 with two 13Cs, so the isotopologues, even though the mass is very closely together. Okay, now I need to drink. Now that we've set up the whole method and we know we can prepare the samples and we can detect a lot of biomarkers, we want to use it for high resolution paleoclimatology. And the best sediments and the best parts the best part to use it in is our high-resolution marine or lacustrine records. This means that they are at best varved sediments or at least laminated sediments, where the preservation of the sediment signal is very strong. We have a good control on the age model and a very high sedimentation rate that allows us for a very high-resolution record. And they are luckily quite a few around all over the world. And today I will give you a very short overview about what we are doing in the Cariaco Basin at the moment in Venezuela. So you see on the side the laminated sediments. And but first I want to start and talk about the Santa Barbara Basin in the United States, and because this is where I did my PhD thesis on. So this is where I know my way around the best. So the Santa Barbara Basin is a very ideal study site, which is why we choose it, because it has varves which are formed and preserved due to this oxygen-depleted bottom waters that we find within the semi-closed basin. So we don't have any bioturbation, and we have a very high sedimentation rate, and that it is composed of this seasonal alternating input from the spring and summer signal, where we have more upwelling, so we get this light biogenic lamina, and during fall and winter we have more rain coming in, and we have a more dark and clastic lamina deposited. And here's the sediment core that I was using, or part of the sediment core, and you can kind of see the laminations and the varves a little bit, but you can even better visualize them by taking an X-ray picture of it, so you see the density differences between these laminations. And this is how we also got a good age control on it, by counting these varves. Another reason why we chose the Santa Barbara Basin is also that it's a very interesting site in terms of Pacific climate variability, because the Santa Barbara Basin is also quite strongly affected by El Niño Southern Oscillation, but also the Pacific Decadal Oscillation. And especially in terms of the current climate change, it becomes more and more important to understand how these cycles behaved in the past and how they probably will evolve in the future in terms of big flooding events or droughts and all these effects that this has, again, on the fishery and on the whole industry in this part. And the third reason why we chose the Santa Barbara Basin is the small yellow dot next to the core location, because the Cariaco organization was monitoring this whole region since about the 1950s, and since 1984 we have seasonally measured instrumental data throughout the whole water column, for example for oxygen, but also temperature and nutrients. So we have a nice data set that we can use to directly compare and evaluate our results from the proxy data too. Okay, so in the Santa Barbara Basin, I did molecular stratigraphy with two proxies, and one of them is based on the haptophyte algae. It's the UK37, and it's based on these long chain alkenones. And the basic idea of this proxy is that they regulate the degree of unsaturation with temperature. So this means when we have colder temperature, we have more of the alkenone with 3 double bonds, whereas when we have warmer temperature, we have more of the alkenone with 2 double bonds. And this can be expressed in this UK37 index, which is nicely correlated to the mean annual sea surface temperature. And the other proxy I was looking at, and the other biomarkers, are the isoprenoidal tetraethers, and those also respond to temperature in the term, or in the mean, that simply said we can find more of the cyclization in the structure with warmer temperatures. And this can also be expressed in a variety of indices, mainly the TEX86, but also the CCAT index, which is based on the major GDGTs, so the one without any rings and the one with 5 rings, the Cariaco. And this is the index I was using. So let's look into some data. And what you see here is the instrumental data. It's not the data I created. So it's the instrumental temperature data from the upper 30 meters of the water column in the Santa Barbara Basin. And when we compare this, so from 1984 to 2009, sorry, just to say this, and when we compare this to our proxy-based record from the alkenone data, we actually see that we have quite a nice match between the instrumental data and the reconstructed data from the UK 37. This is in a monthly resolution, all these data points here. This is actually the instrumental data as a quarterly resolution. So we get a higher resolution with our reconstructed data. And we nicely see that we have this annual cycle in there, and the minima and the maxima and the minima quite nicely matching the one from the instrumental data. But when we take a closer look into it and we want to see how reliable are the alkanome-based temperatures, we actually see that the temperature range is a little bit off, so to say. So we have a temperature range from about 10 to 20 degrees in the instrumental data, whereas the UK 37, the reconstructed data, only has a temperature range from 12.5 to 16 degrees Celsius. So to investigate this, we have to go one step back into the culture-based studies. So we know that alkanones respond to temperature, but there's also a study from Prahl and colleagues that showed that there might be a nutrient effect. So they were growing a culture at 15 degrees Celsius. And what they saw is that when you look here at nitrate and the UK 37, is that when nitrate is consumed over time, the UK 37 is decreasing too, even though the temperature was stable for the whole time of the incubation. And luckily, we have nitrate data, of course, also for the Santa Barbara Basin to compare our results to. And what you see here is the UK 37, our UK 37 temperature data, plotted against the instrumental temperature data. And the gray line here is the one-to-one line. So ideally, if we have a perfect fit, it would all be on the gray line. But we see that we have temperatures that are colder and that are warmer and that are colder than expected. And when we express this as a residual temperature over here and compare this to the nitrate concentration, we can actually see that we have temperatures colder than expected when we have low nitrate concentrations, whereas we have temperatures warmer than expected when we have higher nitrate concentrations. So when we compare this residual temperature, there it is, compare this temperature to the nitrate concentration, we were actually able to see that there's a threshold value of about 2 micromoles per liter that is explaining the deviations between these warmer than expected and colder than expected temperatures of the UK 37. So here for the first time, we can actually say, okay, the UK 37 is nicely recording the sea surface temperature variation. But for the first time in an environmental sample, we can show that this nutrient effect dampens this absolute amplitude of the UK 37 signal. Okay, so now we got a good understanding on the UK 37 signal. And how does the CCAT signal look, the one from the tarmac here, or supposedly tarmac here? So we see already immediately that this signal is not as highly in the frequency as the UK 37. So we don't have this strong annual signal in there. We more or less have longer amplitudes of maxima and minima and a more smoothed signal. So what does this actually mean? And for the signal, I want to say it's not only in the CCAT, so it's also in the ring index when we do it for the GDGTs with the 1 and up to 3 rings. So it's not an issue of the proxy we're using. So we know that the CCAT is sensitive to sea surface temperature. And but we already know and see that this calibration is not as strongly as the one from the alkanones. So we also know from culture studies that there might be a nutrient dependency. And this is a figure from a study of Thomas Evans in 2018. He was still in our lab when he was doing this. And he was growing a culture of N. maritimus, so Taumakia culture, at a temperature that corresponds to about 0.7 in the CCAT. And what he observed is that when the culture was grown with a high ammonia supply, so the blue dots, we have a much lower than expected CCAT value. Whereas when we have a low ammonia supply, the CCAT values are more or less fitting or a little bit higher than expected. So when we look now into our nutrient distribution here with phosphate of the Santa Barbara Basin, what we saw when we just looked at this plot was that our CCAT, when we just reverse it, kind of mirrors this distribution of the subsulfate phosphate distribution. And the same was true for the oxygen distribution. And we not only looked at it by eye and by this visual comparison, so we systematically compared different depth intervals and did a correlation analysis and found actually a negative correlation with phosphate for the whole water column, and for oxygen also for almost the whole water column, a positive correlation. So what does this tell us now for the CCAT signal in the Santa Barbara Basin? So when we look into the Santa Barbara Basin, we know that it's affected by upwelling, and this means that during upwelling, we increase the nutrients, whereas during upwelling, we decrease the oxygen, since to these oxygen-depleted bottom waters. So the response of the CCAT in this study is that when we increase our nutrients, we decrease the CCAT, and for the oxygen, when we decrease the oxygen, we decrease our CCAT value, and vice versa too. But to consider also the results from culture studies, we have to, or we can actually say that for the nutrients, we have a good fit to the one that we see in the CCAT. So from the reported physiological response is that when we increase nutrients, we decrease the CCAT value, and the other way around too. For the oxygen, however, it has been reported in one study that when we decrease the oxygen concentration, we would increase the CCAT. And taking this reported physiological response also into account, we will say that especially in the Santa Barbara Basin, what we see in oxygen and the CCAT is an indirect effect, and it's reflecting this large-scale multi-year upwelling variability that is also responsible for the nutrient distribution in the Santa Barbara Basin. So for the CCAT, we can actually say that in the Santa Barbara Basin, the main environmental driver of this proxy is upwelling and nutrient availability. So to sum it up, for the Santa Barbara Basin study, we can say that with mass spectrometry imaging, we were able to provide new insights into the factors controlling those proxy signals. Meaning for the UK, we can quite confidently say that it's recording the sea surface temperature variability even at this very high, so this monthly resolution. But, and this is the interesting part, for the first time we can show that this absolute reconstructed temperature is dampened by this nutrient effect. And for the CCAT signal, we can say it's reflecting this large-scale, non-seasonal, multi-year upwelling variability, much rather than anything connected to sea surface temperature. And this study is now also published in open access, as you see here. So if you want to get into more detail into this, you can read up this paper too. So another part in the Santa Barbara Basin, another recent study we are working on, and this is not published yet, I can say, is that we want to use mass spectrometry imaging for wildfire reconstruction. So meaning we want to look and see whether we can find these monosaccharide anhydrates, levoglucosan and the isomers manosan and galactosan, and they are all indicative of biomass burning. And the idea is, or the reason why we do this, is that in this region of California in general, and Santa Barbara too, we have quite regularly occurring wildfires. And you see in the map here in red, the whole area that has been burned since the 1950s in this region. And the fires occur almost every year, and they can have, of course, anthropogenic reasons because people are not careful and make fires in regions where they shouldn't, but they are also due to natural occurring reasons, and these are mainly the Santa Ana winds that come from the desert and bring in this very dry and hot air that leads to these fires in this region. And the idea is that with these winds, automatically the monosaccharide anhydrates are transported into the sediment, and we can find them in the sediment core, and that we can detect them with our approach of mass spectrometry imaging. And when we were looking for them into the sediment, we were actually able to detect the monosaccharide anhydrates. And you see in gray the monthly signal of the monosaccharide anhydrates from 1900 to 2009, and in red is the annual running average. And the very interesting part for us was that when we compare this to the area that was burned during this time, we can actually see quite a nice fit of the total area that has been burned here in acres since the 1950s compared to our annual signal of the monosaccharide anhydrates, with this one exception to my finding being here around 1998, 99, which actually corresponds to this very strong El Niño event that led to a lot of flooding and a lot of rain in this area. So the whole depositional process with like transporting sediment into the basin might have been off or might have been different during this time. But we need to investigate this more, and this is all in preparation and not finalized yet. But one major issue was for us, or one major idea was for us, do we really detect the fire biomarkers, or do we maybe produce fire biomarkers? Because with the impact of the laser, it's actually quite hot. So it might be that we have a pyrolysis effect of cellulose to levoglucosan that we just create by shooting with the laser on the sediment. And to simply test this, whether we did a very simple experimental setup to see if we increase the monosaccharide anhydride ionization due to the pyrolysis of cellulose, and also to see if we can detect some cellulose signal itself in the sediment. And for this, we just choose some pure sediment, and then we infused the sediment with cellulose, which would be in green, and then we were shooting with a laser onto the sediment, so doing mass spectrometry imaging with increasing laser power. And what we saw is actually over here. So you can see the intensity of the monosaccharide anhydrates and with the increasing laser power. And we see when we work with about 40% laser power, the signal of the sediment without any cellulose and the one with the infused cellulose are about similar, maybe a little bit higher for the one with cellulose. But when we increase the laser power, we cannot really detect the monosaccharide anhydrates in the sediment itself, because we get out of the range where we can nicely or where we can ionize ideally all the compounds. But it starts that we increase the detection of monosaccharide anhydrates in the sediment where we added the cellulose. So we have to say, actually, yes, there might be this effect of pyrolysis of cellulose that would increase our monosaccharide anhydrate signal. But we also were looking just into the sediment and see if we can detect cellulose. And even with low laser power up to very high laser power, we couldn't really detect any cellulose in the sediment where we didn't add it. So this means that in the sediment where we detected this monosaccharide anhydrates with 40% laser power, this really originated from the compound and not from the sediment and not from the cellulose itself. So when we look into the Santa Barbara Basin, and we're looking for the cellulose, we actually could detect a little bit of cellulose all over the place, which was a little sad news for us. But when we compare this curve to the one of a monosaccharide anhydrate signal, we can actually say that they don't seem to be related. And we use very low laser power to detect these monosaccharide anhydrates. So we are very optimistic that we can use this approach to actually reconstruct wildfire with mass spectrometry imaging. But as I said, this is all still in preparation and not a finalized story yet, and there will be more experiments to come. And hopefully I will have more to present on this, or I definitely will present more on this during the IMOC conference. And I don't know how many people from Brazil will join, but it's all online. And I'm in the afternoon session, so it's just in the morning and not during the night for you guys. And it's a small poster I will present over there. And I would be happy to discuss more on this topic with you on this poster. So as I said, I will give you a short overview of what we are doing at the moment in the Cariaco Basin. And this is where Lars Würmer is working on at the moment. And just to give you an overview of the Cariaco Basin, we also find laminated sediments due to the stratification given by the strong thermocline in the basin and also a deeper chemocline. So we have euxenia in the bottom water, and this creates continuous laminations since about the past 14,500 years. And the laminations are composed of a of a dark detrital lamina, which comes during the warm and rainy summer season when we have this intertropical convergence zone, so the ITCZ directly over the basin, so we have a lot of rain over here. And during the cool and dry winter season, this ITCZ is shifted southwards, and we have strong northeasterly trade winds over the basin, and we get this production of this light biogenic lamina. And already this different color of the sediment can provide some detail or some information on past changes in the Cariaco Basin. And this is a study by De Plazes and colleagues in 2013, where you see the reflectance of the sediment in the Cariaco Basin here in this red line, compared to the Data 18.0 record from the North Greenland Ice Core project over here in blue, and also the data from the calcium, so from the dust input, so to say, from this ice core data. And you can nicely see that when we come from the last glacier, we go to this Berling-Allerod, and then again into this cooling phase of the Younger Dryas, and then in the Holocene, and the reflectance, so the tropical signal is nicely covering and capturing this signal too. So Lars is wants to investigate the changes of the sea surface temperature during the Younger Dryas Holocene transition. And I can so far only give you a small teaser on this because this is very close to submission, and I was not allowed, and I cannot just show all the final figures here that will be published soon, since this is now on YouTube and will probably stay on YouTube for quite a while. So what I can tell you that Lars is looking at using the UK 37 index here too. And you can see here, these are the results from the extractions. So these are not the mass spectrometry imaging results for about a 60 centimeter piece covering the Younger Dryas Holocene transition. And in the extraction-based results, you basically see nothing. It's almost a straight line. So, but when we look in it with mass spectrometry imaging, we get a much more detailed picture of what is going on. And we see a lot of high frequency patterns and changes. And this has about an annual resolution. So with this, Lars wants to identify and these abrupt and short-term climate perturbations that we see during this Younger Dryas Holocene transition, and especially explore this tropical sea surface temperature response to this abrupt North Atlantic warming during the Younger Dryas Holocene transition, which seems to be here a little flattened, but I can promise you he used a lot of detailed information to get out a lot of more that is just to the eye by this of what is going on during the Younger Dryas with the UK 37 signal. But as I said, I can only give you this short teaser, and but I can advertise again the IMOC meeting, and where Lars is giving a talk, also in the afternoon, so not too early for you, about this seasonal to decadal sea surface temperature changes during the Younger Dryas Holocene transition in the tropics. And since I'm already at the IMOC meeting, I like to point out that we're not only working in the Santa Barbara Basin and the Cariaco Basin. So Tiffany Napier from our group is working and using this high-resolution approach to paleoclimate on laminated sediments from the Arabian Sea to actually get sub-annual study of sea surface temperature and summer monsoon rainfall in the 20th century. And this will be presented in a poster, also in the afternoon session or not too early. And also Igor Obrecht is having a talk and giving a talk on decadal sea surface temperature variability in the Eastern Mediterranean, so in the subtropics from the Eastern Mediterranean during the last interglacial. Okay, and with this, I actually like already to conclude and say that during the past years, we managed to get a systematic method development that made mass spectrometry imaging accessible to sedimentary archives. And that mass spectrometry imaging was with this implemented as a strong tool for high resolution molecular stratigraphy. And with this high resolution mass spectrometry imaging, biomarkers can be detected in sediments with this unprecedented level of detail. And this allows for the detection of abrupt and short-lived climate transitions that occur at human timescales. So meaning especially in terms of considering what might happen in the future and how we can compare to what happened in the past. We can really give ideas of what happens in decadal or annual timescales on climate change. And also this can give us definitely new insights into the mechanisms of not only sea surface temperature, but all proxy signal formation. And additionally, with this approach, we are also able to deconvolute signals. So meaning not only to look into one signal, but also deconvolute the whole signal, for example, into seasonality, and get so with this a much closer detailed look into what is going on with the proxies. And we further want and are looking into abrupt climate and environmental change in much older times. So, for example, we are working on the Permian Triassic mass extinction event with polycyclic hydrocarbons. And these are samples from the Maishan region, a section in China, and that's a project together with Weisuke Saito from the MIT and from Roger Summons' lab. And we also go really into deep time, so the mid-Proterozoic expansion of the earliest eukaryotes by detecting steroids in the Barney Creek Formation. And this is a project that just started together with Benjamin Nettersheim, who just started in our group. And we not only want to use this approach to sediments, but it's nicely and can be used very nicely to all samples that contain spatially resolved biomarkers. So this means, for example, microbial mats, where we already published a study from Yellowstone National Park, from the microbial mats in the Yellowstone National Parks, where you see here, for example, just a small picture where we looked at the pheophytine distribution, sorry. But also here we looked into intact polar lipids, so not only the core lipids, but we can also detect intact polar lipids with this approach. And also, this is just, this is a PhD study, a student that just started, who's looking into xenophytophores, so these very deep sea foraminifer, and see how the distribution of the biomarkers are within this foraminifer. And also a study that I will hopefully be conducting soon is to look into geotraps. So this means we can, Morten Iversen, also from the MARUM and from the Alfred Wegener Institute, is collecting sediment trap samples from the water column and is directly collecting the particles in a gel in these trap samples. So we can then look into the particles and actually see or try to see what the biomarker distribution is within the particles in the water column. And with this, I like to thank you all for listening, and I hope this all worked, because it's a little bit weird. I only see my screen. I don't know if I'm completely lost and if everybody else is still listening. But I think it all worked out very nice, and very like a big thank you to the organizers of the Alago webinars, especially Vinicius again. And also to Lars Würmer and Professor Kai-Uwe Hinrichs, who are supporting me with the study. And of course, the whole Hinrichs lab, which you see here on the picture, our whole group, which changed now a little bit because due to Corona and the whole situation, this picture is a bit older now. And also I want to thank Anne Schimmelmann, who provided all the samples for the Santa Barbara Basin and all the knowledge and insights into Santa Barbara Basin sediments. And of course, Bruker Daltonic at this point, because, the machine is from Bruker. They are just across the street in Bremen, which is the perfect setup to start with a big machine like this. So whenever we had issues in the first years, we can just walk over and ask them, and they can help us very quickly. And of course, the funding again, so the European Research Council for funding this whole zoomolecular project. Thank you.
Hi, Susanne. Thank you for your nice talk. Very, very nice talk. I was impressive because you live on the paradise for paleoclimate researcher. You know, we have XRF, high resolution data, but you show us organic compounds high resolution data. So it's really, really impressive. And there is a lot of questions, plenty of questions, and I'd like to address some of you.
Okay, yeah.
Yeah.
I just see the chat for the first time now. Yeah, a lot of questions.
Okay, the first one is from Igor Venancio, and he would like to know what are the main limitations of the MIS methods besides the size of the sample slice?
The main limitation to us is usually concentration of the compounds. So we noticed that when we have samples with, in general, already a very low TOC content and a very low amount of biomarkers in there, it's very hard to detect the biomarkers. This is usually in these laminated and varved sediments, it's usually not an issue because there's a lot of sedimentation, there's a lot of organic material, so there's usually a lot of compounds in there. But we also looked into sediments where we just couldn't detect any biomarkers at all, and then we extracted them and we immediately saw, okay, this is only a few nanograms so only a few nanograms of per gram of GDGTs in there, and we need at least 100 to better 1 milligram of GDGTs in the sediment to get a good signal. And the other limitation is also a bit the kind of compound to detect. So it's the more polar the compound, we have a feeling, the better it works. So we have still some issues with detecting analcanes, because I think it's just this very long, boring structure. There are no functional groups on it. It's very hard to ionize them. But with the fatty acids, for example, as long as we have these functional groups, they are more easily to ionize.
Mm-hmm. Yeah, I should mention there is a lot of congratulations for your presentation.
Okay, thank you.
To go to the second question is from Professor Deborah Azevedo, and she would like to know how do you quantify the compounds and how can you compare between cores or slices?
So we cannot quantify in this term. So we cannot really say it's a concentration. So we just get our signal and we get the intensity. But this is why we work with ratios in the end, to get an idea on the distribution of the biomarkers. But what we saw is that it's when we always compare it to this conventional approach. So we usually always also take samples and extract them again and compare our mass spectrometry imaging result to about at least the average of the sample, what would be in the extraction-based result, because we still, it's a new method. We still always need to double check and prove if it's really true what we see. I hope this was answering the question. So quantification is in this term not possible with so we cannot give an idea about the concentration. We can say a little bit, okay, when we have a higher signal, it's corresponding to a higher concentration. But there are also other reasons like the sediment structure itself, and there might be some issue or some connection to the minerals that are in the sediments. So we have some sediments where we just see it's a very strong signal, but we have other sediments where it's a little bit harder to get out the signal.
Mm-hmm. Yeah. You generally use counts instead of concentration. Yeah.
Yeah.
Yeah. And Vinicius, our colleague, he's also impressive about your high resolution. And then he is asking which kind of dating methods you should apply for this very high resolution that we are showing us.
For the date, okay. Since we are, so it depends on where you are, meaning, so with the Santa Barbara Basin, we have a very good age control on the VAV counting and together with 14C measurements. So we usually use this one. Otherwise, I think it's usually 14C and so far what I used was always 14C and VAV counting, and then also comparing it with and doing the fine tuning of the H model with XRF scanning, so that we can compare it to other cores and really tune some counts to it to get a very strong H control on it. Yeah, but this is. You can also sometimes use, we haven't done it yet, but people from that work with corals were actually suggesting us just to use the alkinone data also as an age control, as we see this annual trend and this annual signal in the alkinones, and they do it actually with temperatures in the corals to correlate this to the age model. But we haven't done it yet. We were always more conservative in saying we need our VAF counts and our 14C ages, and we don't want to make a circular connection of the alkinones to the age again.
Yeah. The next question comes from Daniele Miller from Universidade de Goiás, and she said that it's very interesting, your talk, and she would like to know which kind of metrics are you using for analysis and which program is using to image processing.
So for the matrix, we sometimes use no matrix at all. So for the Santa Barbara Basin, I didn't use any matrix at all. It's all ionizing, or most of it is ionizing as sodium adducts. So we think that the rest of the sea salt just basically in there when we dry these sediments is working as a matrix and is helping the ionization process. Otherwise, in this one study from last Verma, so the first one, they were testing different matrices, and we have these very conventional matrices that are used in mass spectrometry imaging, which are just small acids, so DHB, for example, and they work and they help the ionization process. But we had best results actually with more unconventional matrices such as fullerite, so which is just some graphite basically, or some graphite powder actually too, and then this fullerite. Also some silver, so adding some silver to the sample help too. But mainly at the moment we use no matrix or fullerite, which is kind of similar than just increasing the amount of organic carbon in the sample.
Okay. One more question from Professor Deborah.
Wait,
she would like to know. Okay.
I didn't answer the programming, which program we use.
Sorry. Okay. Sorry.
So Bruca also comes with some programs that we were able to use to get the imaging results, but in the end we used a combination of the software from Bruca, but mainly just to export everything, and then we did it with MATLAB and with some programming. So it's R or MATLAB in the end, and use the imaging tools that are available from these programs.
Okay. So again, Professor Deborah, she has a second questions about which is the required mass spectrometry resolution power.
Now I have to think about what these numbers and how these work again. So we usually with 1 M, so 1 mega. Oh, I'm sorry, I'm not good in answering these questions.
No, you can answer. You can send email and then you can answer later.
Yeah.
No worries.
I'm a geochemist or study geology. I'm not really a chemist by training. Sorry. But it's quite high resolution. It's an FTICRMS we have a 7 Tesla FTICRMS over there, so quite high resolution already.
Yeah, we have another methodological questions, but I think you can go. It's from Boniek. He wants congratulations first for you, and then he would like to know how do you guarantee the integrity of the samples during the slicing process?
Simply be very careful. And to test it further, we use also the microXRF to test it further. So we have our sample before the embedding process. We usually measure everything with a microXRF to get an idea on the elemental distribution. And this microXRF is also giving us a picture. So we kind of see where some structures are in the sediment. And then we can use this microXRF on the slice again. So then we have kind of a quality control if everything is still in place. And if not, we can correct it also with MATLAB and like connecting points again where they originally were at this point. But it's a bit of a delicate work, working with freeze-dried sediments and trying not to break them and embed them. So you always need to be a bit awake and not too shaky when you work with them. But it's working. It's fine.
Yeah, there is a plenty of questions, and I will give you make the last one, and the others will send you by email. But the last one I will mix with mine questions. That's why I would like to make this last one. Douglas and another person is also would like to know, as me, the very interesting relationship between nitrogen and alkanon that you show us. Do you believe that this relationship is related to the migration of Emiliana on the water column?
I wouldn't really. Yeah, it's a good question, because I mean they saw it also in so they saw it in this culture study too, that it's related to this, and from studies from the water column in the Santa Barbara Basin, we actually know that they are mainly within the upper 30 meters of the water column, and they were not found that much deeper. So I wouldn't really. So when it was a study from Robert Tunnell over a few like he did it over a few months, I think, like always checking the water column and looking for these alkanones, and they were quite stable in the water column. So I don't know if they moved that much to adapt to the nitrate, and I think that the upwelling is much faster than what the alkanones or what the Cocolito force can move. So I think they are more they just have to deal with the differences in the nitrate concentration. This would be my suggestion. But I'm not an expert on Cocolito force in this matter. So
Yeah, but very, very, very interesting results. Very important also. Yeah? So Susanne, we'd like to thank you again for your talk. It's an impressive talk. It's so good. And I also need to invite the audience for our next webinar that will be on the next Tuesday, 29 June. But it will be done by Dr. Cristiane Oliveira, talking about the isolation and enrichment of geochemical biomarkers and crude oils and condensates for compounds specific isotopic analysis. So everybody is invited to be there with us, including you, Susanne. I hope you can see you again with us. Thank you so much, and bye, guys.
Okay. Bye.
Bye.